Bismillahirrahmanirrahim. Assalamu alaikum. How are you, dear students? I hope you all are doing well. Dear students, I welcome you once again in Pakistan International School Taif's virtual learning classes. And here we are gonna study biology for first year. Dear students, in this lecture number 34, we are gonna study about viruses as we have started this topic in the previous lecture that and the chapter 5 that was about acellular life. In acellular life, first of all, we will discuss about viruses which we have started in the previous lecture. We have studied viruses discovery, its characteristics, and history. Today we will see its classification and structure. So let's start. Viruses. Viruses and the study of viruses is called virology, and it means poison. That is a Latin word which means venom or poison. Definitely it infests and infect the living organisms drastically. So we will see classification of viruses. Like viruses are classified either based upon host organism or on other structural characteristics, for example morphology, genome type, and mode of action in the host. The internationally agreed system of virus classified is based on the structure and composition of virus particle. That virus particle that is called virion, and mode of replication is also very important in the classification. Mode of replication is seen like in specific classification of we can say which are having DNA, RNA, what type of DNA, RNA are having in their center. Okay, so classification of a virus, if we just talk about based on host. Number first, classification is based on host, and in that we are gonna see some viruses like bacteriophage viruses because it attacks on bacteria, plant viruses, and some animal viruses. So in classification, first of all we will see classification on the basis of their host, and number two we will see classification on their we can say mode of replication and genome type. So these are two very important classifications. So if we see bacteriophage that is actually the virus, and it's the number one category of basis on the host, also known simply as phage virus that attacks and infests only bacteria. The infection may or may not lead to the death of bacteria. When we just see its life cycle, then we will see that there are two types of life cycle in which the bacteria is completely die and some will progress with the viruses. What are these? Lytic and lysogenic cycle we will talk about in the next lectures. So each bacteriophage is specific to one form of bacteria, not on all of type of bacteria, like they are host specific as well. Now we will see plant viruses, like plant viruses, or these are also called viroids. So they are similar in morphology and nucleic acid types to animal viruses, and there are almost 2000 types of plant viruses. And like yet it is discovered that almost 2000 type of viral diseases are known by the scientists. So most plant viruses discovered till date, including very important, there are TMV, that is tobacco mosaic virus, as well as we are having um, that is called potato yellow dwarf virus. So they act uncommonly. Bean mosaic virus is there, bone tumor viruses, they act on corn and sugar cane, and potato yellow dwarf virus we have talked about here. And then they must penetrate cell wall by wounds and parasites, for example aphids that eat sap of the plants, that small insect which sits on that and then penetrate these viruses by just secreting or destroying the cell walls of the plants. So they result like color change, and we can say the deformed stent growth and wilting of the plants can be done by these type of viruses. Now we will see animal viruses. So animal viruses occur as parasites in animals, and there are lots of diseases which are caused by this. They are self-replicating intracellular parasites and completely rely on host animal cell for reproduction. So they use host cellular components to replicate, then leaves the host cell to infect other cells. So that happens not only in human beings but in the cattle, in other different animals. They do for reproduce. So animal viruses, we can see there are lots of viruses over there. For example, viruses cause we can see here foot and mouth disease in cattle or livestock. Rose sarcoma virus cause cancer in animal cells, cancer in animal cells, and many viral infections that attack and destroy the different cells in human body. They cause, for example, mm, papovirus. They cause warts, poxyvirus. They cause smallpox. Almost smallpox is like that is completely eradicated now from our like earth. And then we are having very important that is called picovirus, and they cause polio, hepatitis A, etc. And paramyxovirus, they cause measles and mumps in human beings. So not human beings are affected, but other cattle are also affected by these type of viruses. So but they are host specific. The cattle cannot transmit these parasites or these viruses to the human beings, and like that. Okay, so now we will see a classification that is known as Baltimore classification, and that Baltimore classification that was on the basis of mode of replication and genome type. So that is almost seven groups, and then a Nobel Prize was given to David Baltimore for this classification which is based on structure. So that is based upon the structure, so that we can say about the structure. And in structure we will take genome. So on the basis of genome, some are having nucleic acids which is like of different kinds. Messenger RNA transcription can be done by that, and then we know that after that protein synthesis occurs. So in group 1 we can have dsDNA, like double stranded DNA viruses. These are double stranded DNA viruses are there, and by these viruses, okay, they are having double stranded DNA like helical structure the human beings are having. So they are, for example, poxy virus, and what they cause smallpox virus, and cowpox viruses are there, so they cause pox disease. Single stranded DNA viruses, so they are parovirus, and these parovirus are just like cause rashes on the body. Then double stranded RNA viruses are there, and its example is reovirus. And this reovirus um, causes diarrhea in human beings. Then we are having another group that is called a single, we can say it a single stranded RNA viruses. And these single stranded RNA viruses, they serves as messenger RNA, and they are togo viruses, and they cause rubella diseases, and the rubella virus is type of toga virus. Then group 5, there is single stranded RNA viruses again, but they act as a template for messenger RNA synthesis, and its example is orthomyxovirus, that is influenza virus. Then we are having single stranded RNA again for DNA synthesis, that is reverse transcribing viruses, or they are called retroviruses. We are having here HIV, human immunodeficiency syndrome, that is caused by that, and that virus is a retrovirus. So here we will see group 1 DNA viruses, double stranded, so messenger RNA synthesis on double stranded DNA genome template. And as we know that this type of like this is the DNA which human beings are having, so that is very easy for that to just having control on human body. So there's adenovirus, herpes viruses are there, papo and poxviruses are there. Number 2, there is single stranded DNA viruses. Paroviruses are there, they cause mild rashes. And paroviruses are linear average genome size of 5,000 nucleotides only, and they're the smallest with 18 to 28 nanometer in diameter. So these viral capsids made up of 2 to 4 protein form naked icosahedral structure. As you can see here, icosahedral structure, um, Icosahedral means almost 20 sides it is having, and it can cause like human parovirus B19 disease, arrhythmia, infectious and joint disease syndrome in human beings. Then we are having group number 3, that is a single stranded RNA, in which there is very important coronavirus is there, like that can also cause influenza, and that is enveloped and helical and 60 to 200 nanometer size diameter actually, and the genome's size is 16 to 20 kb. This one is genome. So it replicates in cytoplasm diseases, respiratory syndrome, severe acute respiratory syndrome. It causes. So as like you can understand by this COVID-19 situation that what type of it is. Then we are having group number 4, ssRNA virus, again single stranded RNA, and in which rhabdoviruses are there. So they envelop with spikes, you can see around them these spikes, and made up by glycoproteins. Linked matrix protein contain nucleocapsids, and mumps and rabies viruses can be these are, and they cause mumps and rabies. So orthomexovirus, that is actually influenza virus, uh, that is ssRNA virus, and that acts as a template for messenger RNA synthesis, and that is enveloped, it is having enveloped around it and glycoproteins on that. Okay, where one more category is here about on the basis of capsids. So some viruses, if we just talk about like this, like classification on the basis of capsid. So here we can say the virus is having helical capsids with over all shape of rigid rod like this rigid rod like structure it is, and that is having tobacco mosaic virus is the example of that. Like it appears like this rod like structure, but it is actually this helical structure. So that is on the basis of capsid. Viruses having polyhedral capsids, like it is having lots of shape hadron or angles with glycoprotein spikes on that, and at each vertex, like this is vertex, so each vertex it is having the spike. Adenovirus is an example of that, and virus that affects respiratory uh, like mechanism and eyes, intestines, etc. So viruses having an outer envelope studded with glycoprotein spikes, that is influenza virus, that is the circular one, and that is having spikes on that. It means it is also enveloped. So viruses having complex capsids consisting of polyhedral head and a tail apparatus, like we can say this is what type of this one, this is bacteriophage. It is having head and tail and spikes as well, and with the tail it is having basal plates as well as legs to just capture um, any type of host. So this is bacteriophage. So that is classification and the basis of capsid. Now we will see generalized structure of viruses. In that we will see that viruses is composed of three types of components: nucleic acids, capsids, and envelope. Nucleic acid, which is present inside genetic material. Capsid is made up of lots of cells, those are called capsomeres, and that is enclosing DNA, The inside part, that is called core, and outside there is capsid and envelope. Some viruses are having envelope, some are not. There's called enveloped or unenveloped or naked viruses, and this envelope is having spikes that are made up of glycoprotein, which help for attachment on the host cells. So let's talk about its first part, that is called core. So core genetic material, either DNA or RNA. So most RNA is made up of one-stranded nucleotides, most DNA and two strands of nucleotides also. Both DNA and RNA contain into for making proteins. So actual function is to making protein. So as example, they can cause chicken pox, warts containing DNA nucleic acids. So like poxviruses are having these DNA strands. Viruses that cause flu, HIV, and common cold, they are having simple RNA. So you can see the internal core of genetic material. So a virus is not made up of cells. It is the nucleic acid, DNA or RNA surrounded by protein coat. So they are living inside the host, but non-living outside the host. And the virus uses the cells, enzymes, and ribosomes to make DNA and protein. So general structure, now we will see capsids. So first is core part, and then there is capsid. So capsids, so these capsid, or that is called coat, that is called coat. That is the covering of DNA, not for the protection, but it helps the to just save the nucleic acid from endonucleases, which are the enzyme which cut these nucleic acids. So they are having capsids, that is called protein coat, and closer protect their nucleic acids. So the capsid together with nucleic acid, that is called nucleocapsid, but some viruses have external covering, that is called envelope, and some are without envelope, those are called naked, and each capsid is made up of identical protein subunits, those are called capsomeres. So this capsid is made up of lots of same type of cells, protein subunits, those are called capsomeres, as you can see here, enveloped virus. So herpes virus is having one herpes virus that is having 162 capsomeres, and there are two forms of symmetry in virus capsid. When the capsomeres are arranged in 20 triangles, it is called like this, like it is having 20 sides, like we are having in bacteriophage. So that is called icosahedral. And if the capsomere are arranged in a hollow coil-like structure like this, so that appears at a rod shape, that is called helical structure. Okay, so some viruses are having envelope on that. So they acquired when virus leaves host cell. So this envelope is taken from the host cell membrane. So protects the nucleic. It actually protects the nucleic acid of virion in the um, outside environment, and this envelope is having spikes on that, which expose proteins on the outside of the envelope, and they are essential for the attachment of the virus to the host cells. Some don't have envelope, they're called naked, so they compose only of nucleocapsids, like capsid and the nucleic acid. Some are enveloped, they are surrounded by an envelope like this. Now we will see the structure of bacteriophage. So bacteriophage, as we know, that attacks on bacteria only, and there is a head that is actually the complex structure, complex capsid. So like that is having the head, that collar, tail, and there are, we can say, the legs part. So each bacteriophage is specific to one form of bacteria. So you can see here the composition in nucleic acids inside this one, their genome, and modifies bases are there. So that can be DNA in the case of bacteriophage, and then there is protein that is for protection from the infection, that is the protein case over it. So we can see the tail fibers, base plates, tail, contractile sheath. This tail is having contractile sheath for contracting this one. So that is prism like hexagonal structure and head containing DNA and protein coat. Then there is collar, that sheath around that and the tail. There are basal, this is basal plate and tail pins, and then tail fibers. So head tail connector consists of a collar disc and several whiskers like for the movement of this sheath, because when this contract, so then it can penetrate into the bacteria. So there is a central hollow core tube containing um, viral DNA is injected into the host from this into the host from like a head in a hexagonal plate with tail fibers, usually 6 in number and used for attachment of the host. The tail fibers recognize These are tail fibers recognize the receptor sites in the host cell. Then this sheet contracts down, and this basal plate in which this we can say the tail pins are there, that gets down and penetrate into the, for example, this is bacteria, so it penetrates deep into the bacteria. There is lysosome enzyme which secretes from the core tube of tail, and that lysozyme digests this wall of bacteria. Now we will see structure of influenza virus. So you can see there is a circular structure, and on that it is this is enveloped. There is internal antigens matrix and RNP. This is matrix, as you say, matrix protein, and then RNP, that is a specific protein and are used to determine whether that is A, B, or C. So external antigens, these are hemagglutinin, spike hemagglutinin, and neuroaminidase. We can see this one. These two make the spikes on the envelope, and they are subtype and strain specific antigens of influenza A virus. So there are different types of viruses which are distinguished only on the basis of this H and N. H, hemagglutinin spike, and neuraminidase spike, that represents as well as that is a specific type of protein that is called RNP, that also describes the type A, B, or C. So influenza virus category is A, B, and C, and that is composed of a segmented single-stranded DNA. This is single-stranded DNA. Now we'll see structural biology or structure of HIV. HIV, human immunodeficiency virus, that is retrovirus. This retrovirus determines like its diameter 100 to 120 nanometer, and spherical morphology. It is having the cone shaped core is present. Cone shaped core is present and surrounded by lipid matrix. This lipid matrix containing key surface antigens and glycoprotein. So that is actually enveloped, and that is made up of lipid bilayer, this red one, this one. So that is composed of two types of glycoproteins over there. So these glycoproteins are GP, that is GP41 and 120, and these GP of you can see this blue one is GP120, and this the green base of this one, that is GP41. So this viral core contains two copies of genomic RNA, that is reverse RNA, and there are certain enzymes also. These are called reverse transcriptase, integrase, and protease. So G120 and GP120 and transmembrane GP41, they make the spikes, and beneath envelope another protein shell is present, that is made up of, that is matrix protein. It lies between the envelope capsid. So these GP120 and GP41, they are used to attach on the receptors on the cells. T lymphocyte, actually it attacks on the T lymphocytes. So there are integrase, protease, and reverse transcriptase enzyme is used to convert viral RNA genome into viral DNA genome. Then it just joins with T cells, and we can say that DNA. Then integrase enzymes is used to incorporate viral DNA into the host DNA. And protease enzymes are used to break large structural protein into smaller units, and these structural proteins are encoded by 3 out of the 9 viral genes, and then they are used to make capsomeres and other structure of these retrovirus. Okay, dear students, that's it for today. I hope you understood the structures of the viruses. Tomorrow, inshallah, in the next lecture, we will talk about some different behaviors of viruses, or about the nature of the virus, as well as we study some other topics also. All the best. Allah Hafiz.